Thank you, Megan. Thank you, Chris and Katie. This has just been an extraordinary meeting, and seeing all the different perspectives and how everybody has a little bit of overlap and a little bit of new ideas infused from multiple angles has just been really exciting for me. So I am here as a pediatric gastroenterologist, and my route to a meeting like this was a little bit circuitous and really began because I spent much of my formative years living and working in places like this, rural Haiti, and also southern Africa. And it was there that I really saw the devastation that poor nutrition early in life enacted upon young children. And we all know the statistics. Millions of kids die every year from undernutrition. Here's one I haven't seen this meeting yet. 1 out of every 4 children are walking around planet Earth stunted with signs of chronic undernutrition. So why is poor nutrition early in life so bad? Well, it has a host of effects on liver and gastrointestinal and metabolic physiology. And many of these effects really compound whatever illness led to the undernutrition in the first place, and in some cases make it very, very difficult for a child to gain weight and grow appropriately. Some of these effects include liver function impairment. The undernourished liver does not synthesize the things that it should be synthesizing when it's healthy. And some of these things include coagulation proteins, leaving kids at risk for catastrophic bleeding. The undernourished liver also produces low levels of complement, which could contribute to the immune deficiency we see in these kids. Undernourished livers also do not synthesize normal amounts of bile acids. And these are substances that help us digest the fatty content in our diets, and could be one of the reasons that it's so difficult to orally refeed these kids. Undernutrition also has effects on the intestine in terms of slowing gastrointestinal motility. It's a little bit more complex as to the role that that has in the impaired weight gain. And of course, we've talked this conference about the effect of undernutrition during a critical window of early life, the first 1,000 days, and how these kids are at risk decades later for obesity, type 2 diabetes, heart disease, and other metabolic complications. All of these things our laboratory is actively studying. We are focused on mechanisms by which poor nutrition early in life impair a child's gastrointestinal and liver physiology. And in this circle, in this vicious cycle of metabolic effects that occur, you can find gut microbial dysbiosis. And of course, I put that in quotes because dysbiosis is a bit of a nebulous term, but the altered gut microbiome patterns will be the focus for the next 30 minutes today. So in the clinical world, we typically give our conferences by starting with a case. And here's a case. This is a child right around 3 years old, and it doesn't take really any clinical background to know that this child is severely underweight. But as typical for children in these environments, he didn't begin life severely underweight. In fact, he began life right in the middle of his growth curve, which you can see there. And right around the time he was weaned from mother's milk, that growth curve began falling off. And it was perfectly timed with a series of enteric and diarrheal diseases, led to weight loss, and that, of course, in turn, fed into this vicious cycle of immune deficiency and susceptibility to repeated and more severe infections. And so what we typically do in these situations is, if a child can tolerate it, we will give them ready-to-use therapeutic food, as we heard about yesterday. Now, RUTF has done amazing things in terms of reducing the acute mortality and getting kids to gain weight in the short term for undernutrition. However, when you look at some meta-analyses that look at longer-term outcomes, it's really in question whether ready-to-use therapeutic foods are the best thing that we can give these children. So definitely mixed results in multiple meta-analyses that follow kids over time. And so RUTF was given to this child, and he initially did okay, started gaining weight again. But then, due to a combination of other infections, both malaria and measles were acquired right around this time, his weight began to fall once again. And despite intermittent use of RUTF, he presented, with the picture you see here, in severe acute undernutrition. So what I hope everybody takes away from this conference is that the gut microbiome plays an intimate role in this vicious cycle of pathogen ingestion, enteric infections, which may not necessarily include diarrhea. But a sick intestine yields things like damaged inflammation at the mucosa, barrier disruption, and a sick intestine can't absorb the nutrients from the diet that this kid needs to grow and gain weight. And undernutrition in turn leads to impaired immunity and worsened infection and recurrent infections. And it's a cycle that keeps going on and on and on. What I'd like for us to think about here is maybe at some point in the future, could we target the microbiome with various therapies to try to intercept this vicious cycle from occurring? And so the objectives today, 3 things I hope to cover in the next 25 minutes or so. Number 1 is when we talk about the, quote, dysbiosis or the altered microbial populations of undernourished kids, what exactly are we talking about? What are the patterns that we have seen from observational studies that have really gone back for many, many decades? Number 2, what are some of the factors, the dietary, the environmental, and the host factors that shape this altered gut microbiome of undernourished kids? And number 3, what clinical evidence do we have right now, today, supporting the use of microbiome targeting therapies to specifically enhance growth? So a little history. 1958 was one of the earliest studies that I could find in which somebody was really looking at gut microbes in severely undernourished children. This was Professor Smythe working out of Cape Town. And in a small series of 20 children with kwashiorkor, the severe edematous form of undernutrition, Professor Smythe found that there was a surprising number of coliform bacteria that were actually living in the stomach. So in studies that are very hard to replicate nowadays, he performed nasogastric intubation, sucked out the contents, and grew whatever he could on multiple culture media. And it was very, very surprising that bugs that should be living in the large bowel, much more distal in the intestinal tract, were actually cultured from the stomach. Now, no controls were used in this initial study, but very, very similar results were found over the next couple of decades from researchers in Guatemala, Australia, Indonesia, Brazil, and the Gambia. So what there appears to be, there's an altered gut microbiome, and the initial studies, the culture-based studies, because that's all the technology that was available at the time, showed that that coliform microbes and other microbes that typically have no business living in the stomach and the small bowel were what these researchers would culture. Now, these initial studies also looked at small bowel architecture. So again, very difficult studies to perform nowadays. These are severely undernourished but otherwise healthy children. They had duodenal biopsies obtained, and under the microscope, this is what the initial researchers found. This is not a normal intestine. There's lots of inflammation. The villi do not look normal whatsoever. And this very, very sick intestinal mucosa is not able to do its job. It's not able to absorb the nutrients and assimilate the nutrients from the diet. This is part of a condition that we now call environmental enteropathy. And none of these kids had overt diarrhea. These were kids that were small, they were not growing well, and it was actually quite a surprise to some that their intestine looked this abnormal. So fast forward almost 50 years. Now we have high throughput sequencing. And one of the first studies that employed high throughput sequencing in undernourished children came out of Bangladesh. And you don't see rarefaction curves much anymore. Of course, what the x-axis is the depth of sequencing, and the y-axis is the number of unique taxa that turn up the deeper you sequence. And what this initial study showed is that undernourished children had about a 40% reduction in the richness, in the total number of unique taxa in their fecal microbiota. And this study was also confirmed in multiple other sites in years to come. Now, lest you think this is only something specific to undernourished kids in the developing world, there have been other instances that very, very low body weight is associated with decreased microbial diversity. And here's one example. In patients with anorexia nervosa, as you can see on the left, the PCA plots show a very clear separation from their fecal microbes compared to a healthy weight control. And on the right, you see this is once again richness, number of observed taxa, which is decreased in the anorexia nervosa group. And one thing that keeps coming up is in these underweight, undernourished people, Proteobacteria is one of the key phyla, and especially the Enterobacteriaceae and other potential pathogenic groups keep coming up. Once again, these underweight children and adolescents do not have overt diarrhea. They weren't being studied for that reason. So when you look at the relative abundances, and this is in general, every single study looking at underweight versus healthy weight is not going to find the exact same thing, but as a rule, what we find is increased abundance of, once again, these pathogenic genera, Enterobacter, Escherichia, Klebsiella, Shigella. And not all of these kids have overt diarrhea. Most of them do not. Most who had overt diarrhea were excluded from these studies. And at the same time, in these undernourished kids, we see decreased abundance of genera with potentially beneficial microbes. Roseburia, Fecalbacterium, Butyrovibrio, Lactobacillus, Bifidobacterium. And the last slide for this first part is data you've seen yesterday. And Jeff Gordon and colleagues very, very nicely illustrated this concept of gut microbiome maturity. Healthy kids, all healthy kids in the cohorts that they examined seemed to progress, seemed to mature. Their microbiome seemed to mature as the child aged according to a predictable pattern. And in the bottom part of the slide, you can see the child in red who had kwashiorkor. Despite getting a little bump in his microbiome maturity from ready-to-use therapeutic foods, that increase in microbiome maturity was not persistent, as that child eventually lost more weight and that microbiome maturity fell once again. So to summarize the first part, what are the distinct patterns of gut bacterial community configurations? What is this dysbiosis of undernutrition? Well, in general, this dysbiosis is characterized by decreased richness, fewer unique taxa identified. It's characterized by increased abundance of pathogens and potential pathogens, and a decreased abundance of potentially beneficial microbes. And again, in a few nice studies coming out of WashU St. Louis, this dysbiosis also appears to be characterized by delayed microbiome maturation. All right, so that's number one. Number two, and this is where we're going to spend most of our remaining time. What are the dietary, the environmental, the host factors that shape this dysbiosis of child undernutrition? This is a huge topic, and we are really only going to scratch the surface. For anybody really interested in going more depth, I refer you to our recent review article in Gut Microbes. We discuss each and every one of these in detail. I'll just give you some of the highlights right here. This is one topic in particular that really deserves an entire lecture of its own: inflammation. Remember I showed you the altered intestinal histology of those children, biopsies taken from the 70s, showing all that inflammation, those abnormal villi, that piece of environmental enteropathy. So inflammation is a key component to this. And what inflammation does is alter the microbiome by many, many, many potential mechanisms. Here are my favorite three. So number one, inflammation triggers an immune response. And of course, an immune response can modify the concentration of gut microbes by many, many ways. But one way in particular is to secrete antimicrobial peptides. Okay, so much like when we swallow an antibiotic, hopefully the pathogen is targeted, but there's a lot of collateral damage as well. And some of these innate antimicrobial peptides are secreted, and they will also target subsets of microbes that are not pathogens, perhaps contributing to the dysbiosis. Number two, there is a very tightly regulated oxygen gradient in the intestinal lumen. The epithelial cells have to have oxygen to survive. But deeper in the lumen, in the middle of the lumen, it is a virtually oxygen-free zone. So microbes set up camp according to their oxic, microoxic, or anoxic preferences. So the facultative aerobes live at the mucosa, and the strict anaerobes are living deep in the lumen. And of course, inflammation disrupts this whole process. And the gut bacterial communities have to reconfigure. Some die off if they don't like their new oxygen state. Others will thrive when before they were not able to flourish. And number three, inflammation generates reactive oxygen species and reactive nitrogen species. And there are a couple really, really interesting papers showing how some microbes actually have the capacity to perform respiration. And I'll leave the references to you there. It's actually a really, really interesting story, but it does confer a selective advantage for certain microbes when the intestine is inflamed and more oxygen is present. Okay, other factors. We've talked a lot about diet. And in one key study comparing fecal microbes from children from Burkina Faso versus children from Italy. Now, granted, every one of these children was healthy. This was not a malnourished study at all. Malnutrition was excluded. These authors found that in stool from healthy kids from Burkina Faso, there were microbes such as Prevotella and Xylanobacter that actually harbored enzymes that metabolize certain non-digestible dietary components, including cellulose and xylans. Now, what's special about cellulose and xylans? Well, they make up key components of the Burkina Faso diet, including sorghum and millet. Now, this was only a 16S study, so the bacterial genomes were inferred, but having a nice dietary history, like we heard a few talks ago this morning, I think really can extend some of these associations and lead us to hypothesize why we can find differences among fecal microbes between two different sets of people. Here's another example. We talked about this yesterday as well. Breastfed infants compared to formula-fed infants actually have decreased microbial diversity. And remember, diversity consists of both richness and evenness. And what's happening here, as this study in Cell Host and Microbe a couple years ago very nicely showed, is that breastfeeding massively enriches for microbes that can metabolize HMOs. And so it's a very uneven microbiome, therefore a very low diverse microbiome. But I don't think any of us would argue that that lower diverse microbiome is hurting these exclusively breastfed infants. Take this a step further. Not all breast milk is equal. Another amazing study from the Gordon Lab at WashU, authors went to Malawi, and they recruited mothers who had severely stunted infants and mothers who had healthy nutritional status infants. And they examined breast milk from both cohorts. Using mass spec analysis, the authors found that the breast milk from mothers who had stunted infants was actually deficient in multiple classes of human milk oligosaccharides. And one class, the sialylated HMOs, was studied a little bit further. And these authors produced a lot of HMOs. I had to use a bovine model to do it because it's very difficult to isolate from human milk. And when they gave those milk oligosaccharides to a germ-free mouse who was being fed on a prototypical Malawian diet, low protein, low fat, high carb chow, nothing happened. Okay, the mouse had no microbes. It had nothing to feed these milk oligosaccharides. Milk oligosaccharides, for the most part, travel through the human intestinal tract undigested. We don't have the enzymes to do anything with them. So theoretically, the effects of milk oligosaccharides are mediated by the microbes. Then, when these authors transplanted microbes that were isolated from stool from a 6-month-old severely stunted and underweight Malawian infant, and then fed those mice milk oligosaccharides, you see some of the enormous changes in physiology that we saw in one of the talks yesterday. And very nicely, the authors showed this worked not only in gnotobiotic mice, but also in gnotobiotic piglets. So this illustrates that diet can pretty drastically influence the microbiome, and that milk oligosaccharides, at least in this context, require gut microbes to do their thing. Here's one other example of how diet can lead to a gut microbial dysbiosis that can impair health. So this is another brilliant study. The authors fed mice a fiber-free diet. Okay, probably not unlike what many adult Americans are eating right now. You think about Big Macs and French fries, low fiber. This was a fiber-free diet. And the mice obtained a dysbiosis. And this dysbiosis was characterized by an increased abundance of mucolytic bacteria, in particular Akkermansia muciniphila. So Akkermansia likes to chew mucus, and what the authors showed is that that led to a puny, thin mucus layer in the intestines of these animals. And when challenged with an enteric pathogen, Citrobacter, the mice on the fiber-free diet that had the puny, thin mucus layer thanks to Akkermansia got very, very sick. And this really excited our group as well because, as we've shown before, in one of our mouse models, our neonatal mouse models of early undernutrition, we also found a relative expansion of Akkermansia It's not always 50% of the taxa discovered, but it's always there. And we also have decreased microbial diversity, so mimicking a lot of the things that I've been showing you from the human studies. And we also know that Akkermansia, the relative abundance, typically is inversely proportional to body mass index. So as we learned yesterday, obese people have very low levels of Akkermansia, and thin people, and in this case thin mice, have very high levels of Akkermansia. So that led us to speculate, what does this mean for the host? So looking at gene expression of our malnourished mice, what we found is that these mice are spitting out more mucus. MUC2 is the most prevalent mucin found in the mouse gastrointestinal tract, and as you can see, pretty strongly upregulated at the RNA level. It did not translate to the protein level. When we looked with fluorescence, we didn't see any changes to the thickness of the mucus layer, unlike the previous study, which showed a tiny, tiny thin mucus layer thanks to the mucolytic bacterium Akkermansia. So at this point, we still couldn't tell what is causing this dysbiosis. Why is Akkermansia increased? Is it because there's not enough going through the lumen of the intestine? These bacteria are getting hungry, so they're turning to the mucus wall and finding carbon sources there? Or perhaps this increased MUC2 expression is an innate host response to undernutrition. So one very simple way to get at that question is to repeat the experiment in the germ-free state. And what we found is that germ-free mice undergoing the same malnutrition protocol also have about a 3-fold increased expression of MUC2. And we think this leads to a thicker mucus layer. It's actually a tough thing to answer, so we're still working out all those numbers. But what this tells us is that the increased mucin expression appears to be a host response to undernutrition, not a host response to increased and hungry Akkermansia. So our lab uses 3 models of undernutrition. What I just showed you came from what we call the time separation model. We start separating neonates at day of life 5 away from mothers for 12 hours per day. They get to nurse all night long. The controls are non-manipulated. We also, as we've been publishing these models, we've had a lot of feedback from reviewers, and certainly they're concerned about the stress. And as we saw yesterday from Michael, there is quite a bit of stress that comes along with being separated from mother at this early age. So for that reason, we have 2 other early life models of undernutrition. One is a diet-induced model in which we feed a regional basic diet, low protein, low fat, high carb diet, which models the macronutrient composition of what children in some of these parts of the world consume. So low meat, low beans, high rice, plantains, other things. And we feed the control group an isocaloric chow. Now we give this diet to mothers. The mother's breast milk becomes deficient and the pups become stunted and underweight. We look at the 2 pup models at 2 weeks of life. And because there are some phenotypes that you just can't really measure in a 2-week-old mouse, we repeat a lot of these studies in young adults. And we take that regional basic diet pup model, we wean them to the same regional basic diet chow, and we examine the mice at 6 to 8 weeks. And we've timed it up such that all of our models are approximately 25 to 30% underweight at the time of study, except for the young adult females. Females seem to do better with undernutrition, and this is a theme that we see time and time again. So with these models, what have we been able to, have we been able to recapitulate any of the clinical findings that we see in severely undernourished kids? One finding we were interested in is that undernourished children have slower gastrointestinal transit times compared to age-matched healthy children. And this was a single study. You can see large numbers were required to show an effect, but when you, regardless of whether you had diarrhea or not diarrhea, if you were undernourished, you had slower transit. And this was simply measured by having the kids swallow a marker and count how many minutes it took for that marker to come out in the stool. So obviously, crude measurement, but it's something. So in our young adult mouse models, we see the exact same thing. Undernutrition slows gastrointestinal motility. Here we've given a gastric gavage of a non-absorbable red dye, and we counted how many minutes it takes to come out. Females, once again, respond differently than males. And in our case, females had markedly slower transit time, about 40% increased, versus about the 18% increased transit time that we see in males. So gender-specific effects, it's another theme right here. Now, why does this matter? Well, we think clinically, slow transit in undernutrition has been associated with bacterial overgrowth, probably not a good thing, with abdominal distension, constipation, blunted appetite, probably not good things if we're trying to get these kids feeding once again by mouth. But again, the connection is still unclear. So we can dig a little deeper into this dysmotility. And this time we performed an experiment where we gave a FITC conjugated dextran by gastric gavage, and after a set number of minutes, we sacrificed the animal, removed the intestine, and laid out the stomach to cecum, and separated the small bowel into 10 equally sized compartments. And all we did was measure how much fluorescence made it out of the stomach into the various small bowel components. And a couple of the calculations I can give you are gastric emptying, so how much of the dye has left the stomach at the time that we examined the mice. And we can calculate a mean geometric center. How far did the middle of that bolus travel down the intestine? And by either one of these measures, undernutrition slows gastrointestinal motility. It delays gastric emptying, and the mean geometric center of progression of that bolus of fluorescent dye that we gave to the stomach has traveled less of a distance. And what I haven't shown you here is that these undernourished mice actually have shorter small intestines to begin with. So the total distance that this dye has moved is actually even less than what you're seeing here. But interestingly, this only happened in females. Undernutrition in terms of intestinal transit had minimal effects on the young adult males. And in both of the pup models, we see very similar things, a delayed mean geometric center of progression of fluorescently gavaged dye. And you can see there was, when you look at the stomachs, and these are stomachs here on the bottom right, looking at a control mouse pup stomach versus the maternally separated, the timed separation mouse pup stomach, you can see a massive dilation of that separated animal, and it correlates with an increased or a delay of gastric emptying, which you're seeing in the graph on the left. So that's one of the clinical phenotypes we've been able to replicate in the laboratory. What else? Well, the same authors, more than 40 years ago, once again, nasojejunal intubation, which is really hard to do on severely undernourished but otherwise healthy kids nowadays. But in the 70s, they analyzed this fluid, and they found that undernourished kids had severely decreased concentrations of conjugated bile acids in their intestine. So once again, bile acids are things that help us digest, absorb, assimilate the fatty composition of the diet that we eat, including breast milk. And we also think bile acids, because they regulate gut microbial populations, could potentially play a role in this dysbiosis. So we mimicked the same effect in the laboratory, all three of our mouse models. What you're looking at on the right side of the screen is a heat map of fold changes of undernourished animals compared to age-matched control animals. Any number you see that is in blue boxes and less than 1, which is every one of them, indicates that the undernourished animals had less of that particular bile acid species compared to the control animals. In some cases, this decrease was up to 3 orders of magnitude. So our laboratory models, once again, recapitulate what we're finding in kids. And I believe that the decreased bile acids not only have a role in the gut microbial dysbiosis that we see, but also in the inability to orally feed these undernourished children and help them catch up. So what's causing this? Why this decreased bile acid synthesis? So bile acids are made in the liver, and our undernourished mice have very abnormal livers. This is an oil red O stain, which stains fat. This is macrovesicular steatosis. This is also one of the findings of severe undernutrition in children. About 1/3 of them have fatty livers on ultrasound. We mimicked that here in the lab. And looking even closer, electron microscopy. This is a nice, healthy control mouse hepatocyte. The arrows are pointing to peroxisomes. Peroxisomes are key sites of bile acid biosynthesis. When you look at the undernourished animals, these are not normal-looking hepatocytes. You see altered mitochondrial architecture. You see very, very strange regions of dense glycogen deposition versus very, very minimal glycogen deposition. And most importantly, you see no peroxisomes. Peroxisomes in undernourished mouse hepatocytes are virtually wiped out. And once again, because peroxisomes are one of the key sites of bile acid biosynthesis in the liver, we think that the lack of peroxisomes is contributing to the lack of bile acids that we're detecting, and that also has implications for the altered intestinal microbiota, the dysbiosis that we're seeing. So a more direct connection we're still working on. Lots of other factors. That was just scratching the surface as to what may be the cause of this intestinal microbial dysbiosis. And I would be remiss if I didn't at least mention a couple of things on the other side of this vicious cycle, mechanisms by which this dysbiosis may impair weight gain. So in our animals, when we separate these pups from mom with conventional mice that have all the normal gut microbes that colonize conventional mice, we see that the malnourished mice gain a lot less weight up until their 10th postnatal day compared to controls that get to nurse 24/7 with moms. When we repeat the experiment in the germ-free state, germ-free control mice gain just as much weight. They don't seem to miss not having bacteria. But the mice that we separated from mom, the mice that we gave less breast milk to, actually tolerated the undernutrition protocol better than these conventionally raised mice that have intestinal bacteria. So we're still digging into mechanisms, and we're now doing some selective gnotobiotic transfer experiments to really figure out what is it about that conventional microbiome that is so bad for these animals. And so hopefully we'll have some more answers coming soon. Of course, I have to show one graph that Sathish showed yesterday, a really remarkable finding, once again from the Gordon lab, in that germ-free mice that received gut microbes from healthy children from Malawi did okay on the Malawian diet, the low-protein, low-fat, high-carb diet. But another group of germ-free mice that received microbes from a very, very ill Malawian child with kwashiorkor, severe malnutrition, on that same diet, those mice did horribly and had to be rescued with ready-to-use therapeutic foods. Now, I placed up at the top right corner here, diet is key. Because one interesting thing about this paper is that to find this effect, they had to feed these animals the low-protein, low-fat, high-carb diet that was based on the same macronutrient composition that these kids in Malawi were eating. When these mice were initially placed on a normocaloric, normocarb, normoprotein, normofat chow, this effect was not seen. So there's some interaction between host, diet, and microbiome. That's important. You can't have just two. You have to have all three. Here's one more example to further this point. That consumption of a low-protein, low-fat diet, very similar diets, and this is conventionally raised normal animals, leads to a little bit of weight loss, but really not a whole lot in terms of intestinal histopathology. But when you give those mice a combination of 6 microbes that are not necessarily pathogens, 4 Bacteroides, a Parabacteroides, and a non-pathogenic E. coli, on that same diet, those mice do pretty poorly. They lose a lot of weight, and look at that intestinal histopathology. Inflammation, villus blunting, that is environmental enteropathy. And those same 6 non-pathogenic microbes given to mice on a healthy diet did virtually nothing. Minimal weight loss and no intestinal histopathology whatsoever. So once again, host, diet, microbiome, all 3 matter. Okay, so what causes this dysbiosis? Well, we mentioned a lot of things, and I'll refer you to the paper for a more detailed explanation of some of those other ones. How does the dysbiosis impair weight gain? Well, that's even more complicated, and we're still getting into mechanisms, but hopefully in the years ahead, we'll have more answers. So in the final 5 minutes here, I'd like to share some of the clinical evidence. What has been done, and I'm going to share 4 studies in particular that have actually looked at targeting the microbiome in order to specifically enhance growth of undernourished children. Here was the big one. In 2013, New England Journal of Medicine, Mark Maneri and colleagues studied almost 3,000 underweight Malawian kids who had been given ready-to-use therapeutic foods as outpatient treatment for severe undernutrition. There were 3 arms to the study. These kids either got a twice-daily placebo or amoxicillin high dose, or ceftonir, another antibiotic that we use quite frequently in the pediatric population. And the kids that received placebo, compared to either of the antibiotic groups, had an increased relative risk of treatment failure, in other words, losing more weight, not catching up, and also had an increased relative risk of mortality. This study was not replicated in 2 other very, very large studies, also in Africa, one looking at 2,400 children in Niger, and another one looking at nearly 2,000 children in Kenya. Neither one of these studies showed any effect on nutritional recovery or on mortality, despite up to a 12-month follow-up period. And finally, the only truly large randomized controlled trial, enrolling nearly 1,000 Malawian children hospitalized for nutritional rehabilitation, used a symbiotic, which is a combination of a probiotic and a prebiotic. And this particular one was Symbiotic 2000 Forte. You can see the composition written here. And these undernourished kids received Symbiotic Forte for about 33 days median. There was no significant effect on nutritional cure or on any other nutritional outcome. So why? Why did this 4 prebiotic, symbiotic preparation have no effect? Well, my thoughts are, we don't know what the gut microbiome of these kids looked like. We don't know whether any of these 4 microbes were actually missing. And we don't really know whether they needed any of these 4 prebiotics as well, and whether they would work synergistically with the 4 microbes that were given. And once again, none of these 4 landmark studies assessed how treatment versus placebo affected the gut microbiome. Understandable, given the size of the studies. And of course, one thing we have to think about is we did see an effect in the antibiotic trial, but it is probably not worth the risks to give broad-spectrum antibiotics to every undernourished child around the world. Okay, so that's summarizing my final point. We really don't have enough clinical evidence just yet to recommend any particular microbiome targeting therapy, although the preclinical models that I shared with you earlier in the talk do have some pretty incredible promise. So here's my summary slide. This is where I think we need to go. We have an undernourished child who has gut microbial, quote, dysbiosis. And of course, the goal is to restore him or her to a healthy microbiome and then sustain that healthy growth. Well, how do we do that? So we can give an antibiotic. Of course, we can eliminate some of the pathogenic components of that microbiome. So many adverse effects, I don't think we really need to talk about that as a viable, sustainable approach. What about probiotics? The goal is to restore a healthy microbiota. The problem is that with studies nowadays, the species and strain selection is not currently based on really any good mechanistic data. We're guessing, or we're basing our choice on historical reasons. We can try a prebiotic. It does not have to be alive. Prebiotic can enhance the beneficial bacteria and beneficial bacterial functions, but the target for that prebiotic has to be in the host's intestine. If it's not there, you're giving something that is probably going to have no effect. Ultimately, we need diagnostics to figure out who would benefit from which microbiome targeting therapies, and of course, being able to select the specific individual agents to address one child's functional imbalances. And of course, the long-term goal is to prevent the complete falling off of the growth curve that I opened this talk with, and hopefully put these children on a more healthy and sustainable growth trajectory for decades to come. So with that, I have a lot of acknowledgments, lots of support at Texas Children's, and once again, thanks to the organizers. This has been a fantastic meeting.
Yesterday, we talked very briefly about this diversity thing, right? And you mentioned infants who have a very undiverse microbiome and they're still very healthy, which of course is true, but it's also not really, maybe, I don't know, it's not the same as adults, right? And so you showed results, for example, about the no fiber diet, also the decreased diversity, and we've seen several others. I'm also going to show a slide later. So what do you really think about diversity?
I think diversity is one of many ways that we can measure a microbiome. Right now we know that there is not a single bacterium that colonizes all healthy people. There's some debate about well, again, I don't think there's a single bacterium that colonizes every healthy person. So we need to get to function. We talked about that yesterday. We need to start defining healthy in terms of what functions are necessary. But I think that functional definition is a moving target. And what functions someone needs early in life, growth, vitamin synthesis, may not necessarily be the same functions that an older person needs. Maybe anti-inflammatory properties are much more important as we age. So diversity it's just one piece of the whole picture of what is healthy. And in certain circumstances, yes, increase in diversity is probably what we should be shooting for. But I think what this data shows is that we should not be shooting for it just blindly in all patients, because I think we would be doing these exclusively breastfed infants a disservice if we tried to introduce a whole lot of other bacteria that maybe decrease the efficiency with which they were able to grow from their breast milk.
Yeah, I agree with that. But I think that the key of diversity is the ability for it to adapt to many different things. So what you saw yesterday very nicely is that if you keep feeding generations and generations of mice the same diet, I think that's what it was. It kept decreasing and decreasing, and so sort of the I don't know, like the cellular ecological cost, the robustness of the system just decreases because it can't do everything anymore. Some things just disappear. Anyway,
I really enjoyed this talk. One of the questions that I had, it's two-part, one relates to the other. It's interesting with the mouse models that you presented, a lot of those, correct me if I'm wrong, but they're conventional microbiota experiments. And the Akkermansia finding is really interesting because there have been some studies with humanized microbiota where Akkermansia appears to be protective in a Quatreber model of IgA targeting organisms. And there are obviously other studies out there. So where do you sort of fit in some of the human sort of mouse microbes and mouse physiology studies with the human findings? And my second question is, a lot of the illustrative physiological findings in humans that you described seem to be in the 1970s. So I ask you as a pediatric gastroenterologist, where are the opportunities to study more in humans nutrition in this century? You know, like where should we be going with that?
Yeah, excellent question, Satish. Thank you. So I'll answer your second question first regarding opportunities for understanding child undernutrition, environmental enteropathy much better. So currently there are a lot of global collaborations going. There's one, a large one in Pakistan, which is being spearheaded by some folks from University of Virginia in Charlottesville. And this question is now being addressed. It's interesting. You talk to pediatricians and family care physicians in some of these developing countries, and they say, what do you mean? Enteropathy, what is that? We don't know what that is. This child looks good. There's no diarrhea. I mean, yes, he or she is really, really small, but overall looks healthy compared to everybody else that's going around in this resource-poor environment. So the first step is going to be to understand more about these processes. And it's difficult because performing a gastrointestinal biopsies on children typically requires general anesthesia. And many centers in the places that are so susceptible to these conditions, they're not equipped to do that. And of course, there are a lot of ethical concerns for subjecting children to general anesthesia and invasive procedures for research purposes. So what some of these large collaborations have done have been really to partner with some of the sites on the ground in places like Pakistan, empowered the local people, and in part training them things like endoscopy and what should we be looking for and what does enteropathy look like and you know, potential treatments many years down the road. So these studies are being done. We're still, nothing's been published yet, but hopefully in a couple of years you're going to get a lot of rich descriptive data that's coming from some of the children in the places that we need more knowledge. And then to your other question regarding Akkermansia, Akkermansia is a really interesting organism. And you know, in obesity, in which Akkermansia relative abundance is decreased, giving back that probiotic can normalize body weight, blood glucose, lipid profiles, lots of things. And so at least for the obese population, it appears to be a good thing. We don't know what it means in the undernourished population. In fact, you know, our data here show that the bloom in Akkermansia appears to be due to a host effect. Of course, we've got to do some more mechanistic studies to really prove that that it's the increased mucin expression that results from undernutrition that is driving the bloom in Akkermansia. But I would certainly think that it would have a competitive advantage against other non-mucin degraders in the undernourished intestine. So you know, it's possible that Akkermansia can be beneficial for people at both extremes of the nutritional spectrum, both overnourished and undernourished. We have a lot of work still to do.